Good morning, everyone. It's pleasure to welcome you all to this one-day workshop on PCR and Primer Designing with In-Silico Validation, organized by Flavinlabs Pvt. My name is Shivangi Suman, and today I'll be anchoring you through this program. In simple words, PCR is a method used in labs to make millions of copies of small pieces of DNA. It's like photocopying DNA so that we have enough to study or use. To do this, we need special short pieces of DNA called primers, which tell the copying machine, that is PCR, where to start and when to stop. Designing the right primers is very important, and nowadays we use computer tools, that is in silico validation, to check if primers will work properly before doing the actual experiment. This saves time, money, and get accurate results. So, how exactly does it all work? We'll be exploring that in detail throughout this workshop. But before we dive in, let me introduce you to the lab that has organized this free open session for all of you. So, I'll start the presentation. First of all, we'll be discussing the topics that we are going to cover in the workshop. So, the basic things that we will discuss in is PCR techniques and primer designing. That will include the basics of PCR and its applications, step-by-step primer designing. And after that, we'll see the online tools that is used for in silico validation. And all the students, researchers, and life sciences experts can join this session. Hold on for a moment. I'm sharing the screen, the presentation with you so that you can also see that. So yeah, what we'll be discussing that about the workshop that first we'll be discussing with PCR techniques and primer designing. So all the basics of PCR and its application will be discussed. After that, we'll be discussing the step-by-step primer designing, and then all the online tools that will be used for in silico validation. So as we have said, that all the students, researchers, and life sciences professionals can attend this workshop. And at the end of the workshop, along with the theoretical knowledge, you will have some practical knowledge about the PCR designing and the PCR and the primer designing, and about in silico validation. And if you choose to take the certificate, this certificate of participation will help you enhance in your CV. Along with this, we will be sharing the resource material, that is this presentation, the presentation with the students who are attending the session and is taking the certificate. The time duration of workshop is going to be 1.5 to 2 hours. So after that, let me introduce you with the laboratory. So Flavin Labs is a research and development based company, and our main focus is to turn lab innovations into real world solutions. While R&D is at the core of what we do, we also offer training and internship for students to help them build practical skills. Our lab is recognized by Startup India, certified with ISO 9001 2015, and we have received the prestigious BIRAC Big 21 grant for one of our projects. We also collaborate with government institutions to support impactful research and community driven collaborations. So, we offer a lot of services, but since most of our participants today are students, there are certain services that we offer for students and for professionals and scholars we are highlighting here. So for students, we offer summer and winter training programs for 15 to 60 days, 1 to 2 day workshops. After that, we offer 3 to 6 months projects and trainings. So for BTech students or for master students who have 3 to 6 months project in their last semester, they do that with us. We help students in dissertation and thesis. So there are certain students, PhD and master students who are enrolled in full time work, so they don't have time to perform their laboratory experiments or to write their thesis. So we help them and guide them in their laboratory experiments and writing their thesis. And there is one very important program that we offer, that is biotech finishing school program, and this is for final year students or students who have already completed their course. So it's a guaranteed job program. We have partnered with certain industries, and they provide us with the set of skills that they require in their employees, and we help students in acquiring those skills, and after that we help them in getting placed. So it's a very important program for final year students or students who have already completed their course. And for professionals, we offer certain things, that is food and feed testing, water testing, soil testing, as we have discussed earlier, dissertation and thesis, and tissue cultures of banana and papaya plants. If we discuss about training modules, that we offer training modules in molecular biology, microbiome, biotechnology, nanotechnology, mushroom cultivation, cordyceps mushroom cultivation. Again, this is a very important program, and even Government of India is providing grant to people who are setting up cordyceps mushroom cultivation plants. After that, in biochemistry, statistical analysis for life sciences. So once you have performed your experiment in wet lab, now you have some data, and you need your data to make certain sense. And most of the students who does not understand statistics, specifically life sciences students, and they don't have worked well in Excel sheets, so we teach those students about the statistical analysis, so you can present your data better and make a good sense out of your data. After that, we offer training modules in linkage map and QTL identification, herbal product formulation. So we offer herbal product formulation in pharmaceuticals, in cosmetics, and nutraceuticals, all three of these. If we talk about the time duration, so we offer short term programs of 1 to 2 days till 15 days. And then the long term programs, internship and work, internships that we offer in long terms is from 1 month to 6 months. Now, if you want to join our training modules, trainings or workshop or internship, so you can join both in online and in offline mode. So, if you want to join in offline mode, you'll have to visit our laboratory that is present in Deen Dayal Upadhyay Nagar, Sultanpur Road, Arjunganj. If you know about Lulu Mall, it's from 1.5 kilometers from Lulu Mall. And if you want to join online training, internship, or workshop kind of things with us, so you'll be provided with experimental videos, PowerPoint presentations, study materials. Along with this, we'll be arranging live lectures for you, so that we can solve your queries and teach you things in live lecture, so that it's going to be an interactive session, so you gain a practical knowledge even in even if you're doing an internship with us in online mode. Now, what you will receive if you're doing any kind of internship or workshop program or training with us, so you'll be getting a hard and soft copy of the certificate, and for students who do that in offline mode will get welcome training kit. But one of the most important thing that you get, if you do internship program with us, is personalized letter of recommendation. So what is personalized letter of recommendation? For example, you are going for masters or PhD in some institution, or you are applying for a job, and you need letter of recommendation from industry expert. So students who have done training or internship program with us, on their special request, we provide them with personalized letter of recommendation, that yes, this person, we know this person, and he or she is good with their work. So we provide them this personalized letter of recommendation. Along with this, the certificate that we provide to the students, there is a special serial number that is fed in that so on that certificate. And at the end of the certificate, there is email ID of ours is mentioned. So if you are going for a job and you have presented the certificate that's been provided by us, so and your employer wants to check the authentication of the certificate, so they can mail us that serial number, and we cross check that yes, he or she has done internship program or training with us, and that helps in getting your certificate authenticated. So authentication of certificate is really important, and we provide that also for the students. Now, if we talk about this workshop program, so joining and attending this workshop is absolutely free of cost, so all of you please join and attend and learn. But if you want to take certificate, because certificate is really important to enhance your portfolio, so if you want to take certificate, there is certain amount of charges, very minimal amount that you have to pay. So if you want to take digital copy of the certificate, you have to pay rupees 199, and this 199 rupees cover all the taxes and the processing charge that we have to pay. Because suppose if we are generating 500 certificates, we also have to pay certain cover taxes and processing fee and admin charges that we have to pay. And if you want to take hard copy of the certificate, the price is rupees 499, and you will get in, and you are paying rupees 499, so you'll get premium hard and premium laminated hard copy of the certificate, and this certificate will be dispatched to your location, at your home. and why it's worth taking certificate with us. So as we have discussed at the very start that we are an ISO certified laboratory, so that will add a strong credibility to your certificate. Along with this, your certificate will be of premium quality, and the main purpose of certificate is that this will enhance your CV and your portfolio, and you can present somewhere. Of course you have learned things, but to make people believe that you have learned that thing, you'll have to present the certificate. So that will help your CV. And our certificate is globally valid, so you can present that certificate anywhere across the globe. Now this is all about my presentation. So before I'm going to end, I want to say ki all the students who want to take certificate, the registration certificate registration form is already shared in the community. You can go there and fill the form, take screenshot of the payment, and upload that on your in the form section. There is a place where you have to upload that payment screenshot, so do that. And those who have already filled the form, you need not to fill the form, so please do not fill the form again. And all the students who are attending the session, please write your names in the comment section. This will be considered as your attendance. So please do that. And now I would like to invite Sir, Mr. Sandeep Kumar. He is a director and R&D head of Flavinlabs Pvt Limited, and today he'll be taking your lectures. So please welcome Sir, and all of you, please attend the session till the end, and happy learning. Thank you all.
Good morning to everyone. Hope my slide is visible to you. Today we are going to cover basics of PCR, primer designing, and in-silico validation. You all must have, if you are a student of life science, especially biotech or microbiology, then you all must have heard about PCR. Um, you all know that PCR is a very important technique for life sciences. In this presentation, we will be covering introduction and fundamental principles of PCR, its reaction chemistry, its process, PCR variations, how to design a primer that works, and how to validate it in silico, because in silico validation is a very important step because it um, saves us the cost and our time. So first we will start with PCR molecule. PCR, it is a molecular milestone. It is a discovery that changed biology entirely. PCR was invented by Dr. Carey Mullis in 1983. It helps exponential amplification of specific DNA sequence in vitro conditions. Previously, the whenever you want to clone your DNA, you have to go through labor intensive process of gene cloning, and it takes a lot of days. But with the invention of PCR machine, uh, these days turns into hours. For his this Siemenswell discovery, Dr. Karimelis was awarded with Nobel Prize in Chemistry in 1993. It made molecular diagnostic, forensic sciences, pathogen detection, and gene expression study very feasible and accessible. As you can see, I've also represented an image of original PCR. There was three compartment which were temperature controlled, and we have to add manually polymerase in each reaction. We will go through with the short history. In 1983, concept of PCR was demonstrated by Carimeles. In 1985, TAC DNA polymerase was introduced from Thermus aquaticus, which eliminates the need of adding fresh enzymes at every cycle, because in earlier uh, we have to add polymerase in each and every cycle because it was not very temperature stable. In 1990s, the emergence of programmable thermal cycles, which we are familiar with these days, will started. In 2000, real-time PCR or quantitative PCR started, and after the 2010s and 20s, digital PCR emerged, which helps in quantification, absolute quantification, and single molecular resolutions. What are the uses of PCR? As we all gone through COVID, we were very versed that PCR is used for pathogen detection, especially SARS-CoV-2. Apart from that, it helps in many other pathogen detection. In research, it helps with cloning, mutagenesis analysis, site-directed modifications, and gene expression analysis. In forensic and law enforcement, STR profiling for identity and kinship of analysis can be done in very fast way and very precise way. In agriculture, biotechnology, GMO detection, pathogen-resistant gene screening helps us in various ways for designing new crops or getting new varieties. The fundamental PCR is like that it is based on thermal cycle. First, we denature the DNA, in which the hydrogen bonds of DNA breaks, and we get a single standard DNA. As you can see in image, first step is denaturation. In annealing step, which happens in between 50 to 65 degrees centigrade, the primer binds with the complementary target region. As you can see, that pink colors segments are primers. At annealing step, they will get bind to the our target sequences. And third step is extension, where polymerase attaches to the DNA and it increases the length of the product. So extension, it is in simple way you can say it is photocopying our target DNA in each and every cycle. After second cycle, the amplification occurs in exponential way, 2 raised to power and in 2 raised to power n cycle, and another cycle you will get 2 raised to power n copies. So usually this amplification lasts for 30 cycle. We will discuss this later in detail. So, what are the core components which we are required for a PCR? We need a template DNA. Whatever is our target sequence is called as template DNA, and usually 1 to 10 nanogram template DNA is required depending upon the source and condition of our PCR. Second is enzyme, that is called a DNA polymerase. Usually we use tag DNA polymerase, but high fidelity DNA polymerase are also used when precision is required. Tagged DNA polymerase, it catalyzes the DNA strand synthesis. Mostly it is required from zero point 0.5 to 2 unit per 20 to 50 microliter of reactions. Please note that here is we are whatever we are discussing is in microtons, not milliliter. Okay, so it is required for zero point 0.2 to 2 unit per 20 to 50 microliter. We need DNTPs, that is DATP, DTTP, DGTP, and DCTP. These works as a building blocks for DNA, and we require zero point 0.2 millimolar for each reaction. MgCl2 or magnesium chloride, as we all know that our polymerase needs magnesium ion as a cofactor. Without magnesium ion, it will not start polymerization reaction. So we need magnesium chloride for that. Buffer, we in system buffer we also need, because as we know, whenever we are going with biological process, pH plays an important role. So we need buffer. It maintains pH and ionic strength. Its final concentration is 1x. Apart from these template DNA polymerase, DNTP, magnesium chloride, and buffer, we also need additional additives like DMSO, BSA, betaine, 8GC rich templates. So their concentration varies. Why we need them such many times that whenever we isolate DNA from any plant or animal sample, there might be presence of some amount of secondary metabolites or components which are known as PCR inhibitors. So DMSO reduces secondary structure in G series templates. Betaine use whenever we have strong secondary structure, we use betaine, and BSA, what does it, stabilizes enzymes and neutralize the inhibitors. Now we will go through what are the quality considerations for our PCR reaction. Whenever we are going to PCR, because it involves enzymes and chemicals which are costly, and plus we are pouring our time and our labor in it. So we make sure that whatever components we are going to use are optimal for our PCR. So source that DNA should be, it can be from plasmid genomic DNA, or it can be cDNA if we are talking about real-time PCR. Its purity, that is purity measured in terms of absorptions at 260 by 280 ratios. It should be always, not should be, it must be always in between 1.8 to 1.9 for DNA and about 2 for RNA. If it is lower or higher than that, that means we have contamination of protein or secondary metabolites, or our DNA is degraded. Concentration can vary from 1 to 10 nanogram per microliter in case of plasmid. If we are going with genomic DNA, then it can vary from 52 to 50 nanogram. It should, our, whether it is plasmid or genomic DNA, it should be intact without fragmentation. There should be no shearing, and it must be present in high molecular weight. It is preferred that it have high molecular weight. You can run a gel electrophoresis if you, when you are running a gel. After visualization, you can see there will be a sharp intact band should present. After that only you can go for um, PCR. It should be contaminants free. What is contaminants? Like protein, phenol, ethanol, EDTA, or any detergent. They should not present in our DNA or plasmid which we are going to use for PCR. So, what are the primers? Primers are a starting point for our amplification step. Its length should be 18 to 25 nucleotides. Its GC content should range from 40 to 60. Its melting temperature should be 50 to 65 degree. And forward and reverse, difference between temperature of forward and reverse should within 1 to 2 degrees centigrade, so that they can anneal properly. Its 3 prime and stability should be that there should be no runoff 3 more than 3 G or C's to avoid self complementarity. Secondary structure should not be present, and its specificity should be checked via a CBI blast or primer blast. So again, let's start with deep diving in our PCR reaction. On your left right hand, you can see there is a thermal plot which explains each and every step which are going to use in PCR. Whenever we are doing, first step is initial denaturation. In what is initial denaturation means, it occurs at 94 degrees centigrade for 2 to 5 minutes. Sometimes it goes up to 10 minutes also. What it will do, it denatures all the second standard double standard DNA. It converts into single standard DNA due to high temperature. As we all know that DNA, both strand are connected with hydrogen bonds, and hydrogen bonds are temperature level. Whenever you increase the temperature, if it reaches up after a certain threshold, hydrogen bond get disconnected, and your double standard DNA will get single standard. After that, second cycle starts. Second cycle start with denaturation. It composed of three main cycle: denaturation, annealing, and extension. This cycle is also known as amplification cycle, because actual amplification of your DNA is going to be in this cycle only. It repeats, and up to 30 to 40 cycles. Denaturation, after initial denaturation, it enters in denaturation cycle, where again it is at temperature at 94 degrees centigrade. Still it is in single standard. At annealing, your primer get binds with these uh, denatured single standard DNA, and polymerase attached at that same step, and it start attaching DNTPs according to complementation, like A to T and G to C. So it occurs at annealing steps and extension step. Sorry, not annealing, at extension step. Your polymerase attach somewhere in between after annealing and start of extension. And extension, your polymerase will start adding DNTPs at their respective places, and your DNA get replicated. Again, it will go to the nature stage. After the nature stage, it will again break into single standard DNA, and then again primer will come and bind with it, and at extension polymerase again get attached with it and replicate it in multiple copies. This happens for like 30 to 40 cycles, and then it will enter into final extension, wherever all the remaining amplicons will get completed, and then we cool down it to 4 degrees centigrade to prevent additional processing. At the temperature get lower down, your polymerase will stop acting because temperature is very less, and we will get after this we will load it into electrophoresis unit and we absorb the bands. Okay, hope it is clear. I'm not going in very deep because we have time constraint of 1 to 1.5 hour, so we can't go deep down. If we go here, then we can't process this in silico validation process and primer designing step. So whenever you do PCR, we go through. Sometimes we don't get result, and we makes us ponder that where we went wrong. So these are the common issues that we can solve just by thinking and reasoning. If you get no band, then first what you have to do, you have to check your quality of your DNA, your master mix, that whether they were stored in at optimum temperature in -20, they are fresh or they are not very old or expired. And then we check the cycler setup. Sometimes we have some mishap happens in our machine, so it couldn't work properly. So mostly if there is no band present, then we check all this. If we are getting non-specific band, what is non-specific band? Non-specific band means if you know that your PCR product should be 100 or 200 base pairs, for it is 200 base pairs, we know that our band must come near 200, and it goes above like 500, 400 or 1000, or goes below like 100 base pair, then these bands are called non-specific band. To resolve this, we increase annealing temperature and we may reduce primer concentration. If you are getting a smear in your gel electrophoresis result, then you have to use, that means the DNA you have used was very fragmented, and we have to use fresh DNA, or maybe you have gone number of cycles required, more than number of cycles which were required. So we reduce the cycle count. Sometimes we observe primer dimer. Primer dimers are the fuzzy bands which we visualize in PCR below 100 base pair. For to rectify them, we redesign primers, or we reduce quantity of our primers. So now we are moving towards types of PCR, because there are numerous types of PCR. It is not possible to cover all each and every PCR types within 1.5 hours. So I will just mentioned only a few which are relevant to beginners. First is conventionally or endpoint PCR. Whatever we have discussed, that cycle 3 cycle PCR, that 3 step PCR, that denaturation, uh, annealing and extension and then hold, that is called conventional PCR. It is amplification in monitored at the end of, you can only see whatever the product is formed at the end of PCR after visualization using agarose gel electrophoresis with ETBR or cyber safe staining. Okay, you cannot determine what is happened in between. You just get in last. So it is qualitative PCR. It is not quantitative. Qualitative means that you are going to judge on the basis of what you can see or observe with your eyes, not in terms of number. Okay, that like an example, you can see that in image which I have pasted, it shows some have single bands, some have double bands, or some have sharper band and some have little fuzzy band or little less intense band. So on this basis you can correlate our DNA amount or the variation in DNA, but you can't quantify exactly what amount is there or what size is there. You just can approximate everything. So this is conventional PCR. There is another PCR which is widely used these days is real-time PCR or quantitative PCR because it tells the exact quantity of the DNA or copy number of your gene present in the system. So it is known as quantitative PCR or qPCR. Why real-time? Because in real time you can see the number of amplification in happening in form of a graph which is represented by machine. Okay, in this, fluorescence is measured at each cycle, which directly corresponds to the quantity of the copy number of your genes, particular gene, which is your gene of interest. There are two detection chemistries which are used. First is cybergreen, and another one is TACMAN probe. Cybergreen is a dye that intercalates with your double standard DNA. Whenever it intercalates, it emits a signal which is recorded by a recorder and translated in terms of CT value or a graph. In terms of DNA probe, as you can see on your right hand, that DNA probe based real time PCR, there is a probe, DNA probe is used, which is and initially there is a quencher which is represented here in black dot. Okay, so it is quenched by its emission is stopped with help of quencher whenever it get binds with our target DNA, sorry, target cDNA, strand of cDNA. It get quencher release its fluorophore and it emits fluorescence, which again get recorded by detector. The main difference is here that in dye based you need not to design probe, but in DNA probe based you have to design a probe that is complementary to your sense stand of cDNA. Okay, so every whenever it releases a signal, there is a once this signal crosses a certain value, it crosses it, that point is known as CT, that is cycle threshold. It correlates to starting of template quality. It is used for gene expression analysis, pathogen detection, SNP genotyping. It was widely used for detection of amount of COVID viruses present in your system at the time of COVID. Every doctor was suggesting RT-PCR to detect whether you are considered as a highly contagious or less contagious. In real-time PCR, we have to do data analysis with help of softwares, and for that purpose, we have to understand some common terms which are very common for this real-time PCR. First term is CT or cycle threshold. What it denotes? Cycle at which fluorescence cross this background. As you can see is in image, there is a red color line is present. You can see it, and there is a dotted line is your threshold line, that is your baseline. Below this, whatever is happening will not be considered as expression. Once your signal, your gene produce enough signal that it crosses this threshold line, fluorescent enough fluorescent that crosses this line, that means at that cycle your target gene crosses this. So this is denoted as CT. Okay. Once this CT is produced, it is recorded by the machine, and in terms of it is converted into Excel sheet, and you will record this CT. This is called as CT value. From this CT value, you will calculate delta CT. Okay, delta CT. Um, CT. Once you get CT, now in terms of real-time PCR, you always need a reference gene. Reference genes or housekeeping genes. These are those genes which are constantly present in your system or your subject system. So these provide a baseline. There you must be familiar with terms basal level expression, that we all know that genes are certainly always present in basal level express. They are not altering any process, but they are just present there. To normalize that, we need uh, housekeeping gene. So to get delta CT, first we will minus the reference gene CT to the target gene. For example, your target gene CT is 16, your reference gene CT is 25, so you minus 16 minus 25, that will give you value of delta CT. Hope this is clear to you. Then we will go for delta CT. Delta CT will give you the amount of in terms of expression, that whatever the changes in your control sample, and what is the change in your target sample. So you will minus the sample of interest, whatever your sample of interest, of delta CT of sample interest minus delta CT of reference sample. It will give you the delta CT, which will further convert into fold chain. Fold chain will give you the value of expression level, whatever the change in the gene, to control how fold it is higher or how fold it is lower to your control sample. That will define that whether gene is your upregulated or downregulated. For simple terms, for example, you are experimenting with some disease resistance. Okay, so if your particular gene of interest is higher, its delta CT or fold change is higher than your sample or control, then you will say that yes, this particular gene is higher, so it is corresponding to this. This is making the sample, our sample, susceptible to the this particular disease. If it is lower, then you can say that due to less production of this particular enzyme or gene, our sample is getting uh, targeted to the disease. Next version is multiplex PCR. Multiplex PCR is generally used in pathology, plant pathology or any pathology. What we do, we all know that our gene can one disease can be targeted by various gene or various segment of DNA. So we make multiple primers for this segment, and we put in all at once, and then when we done it in PCR. So more than one PCR can be used for more than one primer can be used for single PCR at the same time, but condition must for they must follow that their PCR product should be of different size. Okay, where it is used when we have sample which is rare or which is present in very small amount, or it is we need multiple tests for a confirmation of a single uh, disease. So what we will do, we take multiple sequences, we put in PCR. Whenever we run, we get number of products. For example, if we are saying that for a particular disease, there must be four primer pairs should be all positive. If any of them are absent, then we can see that there must be a variation in this particular strain. It is used in pathogen detection, SNP genotyping, forensic sciences, What is the advantage? It is a very fast and cost effective method and high throughput. But it is very tricky to design primer on that segment that which produce different, each primer produce different molecular weight product. Okay. So plus it due to this multiple weight molecular weight of product, its productivity get sorry sensitivity get reduced. Next one is nested PCR. It is a modification of the standard PCR techniques that significantly enhances sensitivity, specificity, particularly when amplifying DNA target that are present in low abundance. Whatever happen, sometimes we don't get a large amount of DNA for everything. It happens in two rounds. In first round, normal one outer pair, there is a two pair of primers. One is outer pair, another was internal pair or second pair. First pair primer normally just like previous conventional PCR. It happens we add both primer or one by one we first run it and whatever the PCR product generated in first round, they work as a template for second internal primer. Okay. For example, you can say that we have two primers pair, one and B. Okay. First, you run a PCR with conventional PCR, you get a PCR product. Normally, convention PCR, what we do, we run electrophoresis directly. We go for electrophoresis and visualize it. But here, what we will do, you will not be doing electrophoresis. You will save that PCR product, and instead of DNA second round, you will use that PCR product as a template and again do the primer PCR with second pair of inner primers. Okay, so you will get very specific and amplified band with less quantity of DNA. Okay, so it is widely used for pathogen detection, SNP genotyping, and mostly it is used in forensic sciences. In forensic science, we have very limited amount of genetic material available, and we have to work with that. So what we do first, we did photocopy of original DNA, and then we amplified it. Advantage is that it is highly sensitive, highly specific. It improved detection of low abundant target. The disadvantage is it is very time and cost incentive because you are going to do two round of PCR instead of just once. Because of this, the risk of contamination is also happens. Other one is touchdown PCR. Sometimes in our day-to-day life, when we are running PCR, we notice that we everything is fine. Our PCR primer is also okay. Our DNA is also of very good concentrator. Still, we are not getting desired product, or we are getting very artifacts. There are number of artifacts are present. Then we go for touchdown PCR. Here we refine its annealing temperature to get proper band. So we start with higher annealing temperature. For say your annealing temperature should be um, 55. So we increase it to 60, and it gradually decreases temperature 1 degree per cycle until we reach our target annealing temperature. And in annealing, there is a in PCR machine there is a option that helps us touch down PCR, that where you can input cycle that particular per cycle how much ramp rate you want. So when we design new primer and we are not getting a specific band, then we can do this. And whenever there is a GC content variable, region which have variable GC content, you can use that also. So now we are moving to primer designing. So for primer designing, this is a workflow. First, you identify that your target region. For primer designing, one thing you have to make sure that sequence information is very must. If you don't have sequence information with you, you can't design primer. Sometimes you can do, but they are donor degenerate primer, but they are not very specific. So we have to first identify a target region which we are going to amplify. Then we will use primer designing tool to design primers. After designing primer, we will go for primer dimer check and a hairpin check, that is quality check. It is a essential step to ensure that whatever primer you have designed should work. If you skip this step, you can't be sure that your primer will work, and you will lose time and money. Then we can validate its specificity with help of blast. This is an optional step you can do, or you can avoid this, because if you are sure that your target is particular gene, whatever you are aiming for, and you can skip this. Then you send it to for primer to companies order, you will, and you will get in lyophilized from once you get lyophilized primer with you, you resuspend in either tea or water, and then you will go for PCR. So before designing primer or going with designing actual designing step, we have to make sure that it follows certain criteria, so that whatever we design, it is of your optimal use. First, it length. length Length matters. It should be in between 18 to 24 nucleotide for a standard PCR. In some PCR, like if you are going for molecular markers like RAPD, then it can be 10-mer. There 10-mer primer is used for RAPD, but that is a special case. In normal standard PCR, you 18 to 24 nucleotide is enough. Why 18 to 24? Why not less or more than that? Because 18 to 25 is considered as specific enough to bind at your target site. If you are going for less, then there is a chance that your primer may fall off due to temperature changes. Its melting temperature should always in between 52 to 60 degrees centigrade. You can go higher or lower, but ideal temperature is 50 to 62 degrees centigrade. Its GC content should be in range 40 to 60%, but ideally it should be in between 50 to 55. If you, why not more? Because if you go for more higher concentration of GC content, then there is a chances of secondary structure formation, and which leads to non-usability of your primers. We try to add a GC clamp at 3 prime. And what is GC clamp meaning? At the end of your primer, there if one or two GC pair are available, then it is considered as a good primer, because as we all know, GC binds with triple, make triple bonds, so it is provide a strong adherence to your template DNA. Okay, we should avoid secondary structure, hairpin and primary dimers. Hairpins are the structure that your single primer bind with itself and form a hairpin loop structure. And primer dimer means primer uh, can contain sequence which are highly complementary to itself, or reverse primer. So they will bind to itself and they will not be available for annealing step. Okay, repeats and then avoid. We should avoid long runs of single base pair repeats or dinucleotide repeats more than two or four, because they will again leads to hairpin formation or primer dimer formation. Okay. So 3 prime and stability. Uh, Unstable 3 prime and that is less delta G is preferred because it reduces false priming. It avoids complementarity of 2 3 base of 3 prime, and again they avoid secondary structure protection. Sorry, secondary structure formation. We should always avoid that. Okay, now we will be moving to actual hands-on process. If you want, you can just open a Chrome or Edge or whatever browser you are using, and side by side, and you can follow along with me, or you can note down and you do this on your own later on. Okay, and whatever questions you are having till now, you can post in chat box, and we will try to answer as many as we can at the end of this workshop. So this portion is going to purely hands-on from now onwards, whatever we have learned till now. So softwares we are using NCBI for sequence travel. Primer 3 plus we are going to do for primer designing. Oligo analyzer we are going for quality checks. Primer blast we are going to use for specificity check. And for in silico PCR or ePCR, there are three softwares which we can do use, and whenever you have your target in your mind, before going your actual wet lab, you can do the dry lab PCR and get results and in presentable form also. Okay, so some of you might have knowledge of NCBI and sequence retrieval, some might not have. So today I am going to show with this start. Okay, so first for NCBI, you can simply Google NCBI. Okay, this is a homepage of NCBI. Hope I'm not going very fast. And from here, you can search for nucleotide database. It contains detail of all databases, but because we are going to do uh, primer designing, and for primer designing, we need nucleotide sequence only. So we will be doing with nucleotide. Any gene you can search. For example, I am going to do actin gene. If you click here, you will get actin of gene of all the sequences whichever being submitted in this NCBI. Okay, you can go for any particular plant. You can just use it as Google for your sequencer. If I want do for actin of Arabidopsis, I can do search from here, and then you can see that there are lots of Arabidopsis thaliana chromosome number 3 partial sequence, chromosome number 3 Arabidopsis thaliana actin 12, and multiple entries are there. Here you have to look for your particular sequence, whatever query you want. Just like Google, once you do any search in Google, it will give all possible result, because it fragment your query and search according to that. I have done Arabidopsis and actin, so it will search wherever actin Arabidopsis thaliana came and actin word came, because in chromosome 3 there might be actin present, so it is giving that also, but we need only actin. So what we will do, we select actin 12. Okay, now here you can see there is a faster sequence is available. We need this sequence for our primer designing, so we will go directly to faster. We will not discuss much about whatever is present inside this, because we already have covered in our first workshop. So those who have attended are already well versed with what is FASTA format. Again, I will repeat that FASTA format is a specific format to store your nucleotide sequence, so that your computer can understand that it is a nucleotide sequence, not some random gibberish. Okay, so how it is identify that, your first line is known as identify line. Like here you can see this always starts with the greater than symbol, then your identifier line that contains your sequence information. After that, there is a one indent, and after that indent, whatever comes is considered as a sequence. So what we will do, we will copy this sequence from here, or you can download it or paste it in different file, and we will directly move to primer designing. We will use primer 3 plus. Okay, primer 3 is a standard algorithm to use uh, primer designing. Whatever software you have used till date, whatever whether you use primer designing or primer blast, they all work on same algorithm from primer 3. It is derived from primer 3, so we will use primer 3. Okay, just get familiarized with primer 3 plus first, homepage first. So here you see that there is a section of task. Okay, in task you can tell software that whatever you want to do. You want a generic primer designing, or you want cloning primer, or you want discriminating primer, or you want to pick some sequencing primer. Depending upon that, it will change the its basic default settings. It will change, but we are not going to complicate it much. We are going to keep it simple. We just keep it at generic. Okay, then you will see, let me a bit zoom it so that you can get a better view. Hope this is visible to all of you. Kuldeep, check दिख रहा है ना proper? It is visible. Okay, you will, here is a main window. In main window you can paste sequence id whatever you want. If you want to give your sequence a name, you can paste the sequence id. You can upload your own file. You can use an example file, or you can paste your faster sequence like that. Okay, always make sure it should be in uh, faster format. Then you will get some exclusion and exclusive inclusion reason. ठीक? Like there, if you put anything in this pointed bracket, then it will be get excluded that portion. Like for example, if I select this and put these symbols here, then it will get excluded from this region. It will not take it as a target region. It will exclude this region specifically. So it will not design any kind of primer which include this region. If I play square bracket, then it will actively try to include this region in primer so that it comes as a target. Same happens with include. Okay, so it is a very specific use case. It's not necessarily for doing every time, so you can do that. Now we will move to general settings. Okay, as you can see in product size ranges, there are number of ranges is there. You can write as whatever you want. You can just depending upon what your primer size you are targeting for. Normally, in normal cases, we go for 250 to 500 base pair. You can write that only. You can do multiple product size like 200 to 500, then comma, then add 700 to 1000 base pair, whatever you are targeting for. Okay, let's just keep it simple by 250 to 500. Then primer size, as we already discussed, that our primer should be in range of 18 to 25 or 27, whatever you want. Optimum should be 20. 20 base pair is sweet spot which we are going to target. Your primer TM can vary from 57 to 63, but here whatever you put it optimum, the software algorithm will try to first match these criteria like 20, 60, primer bond percent 97, GC percent 50. Okay? And once if it is not there, then it will shift to left or right. Okay, so maximum TM difference is 100. As we was going through the slide, we saw that I've said that maximum temperature should be 2. But why here 100? Because in algorithm term, it is taking 100. You can, it is similar to 3. And good thing about this software that if you are not getting what this particular terms is meant, you can directly hover on this blue color text and click on it. It will open in a new window, and there it will guide you with everything whatever it is telling. Okay, like I have just clicked on what I clicked on TM difference. You can see max TM difference here. It is saying that maximal acceptable unassigned difference between melting temperature for the left and right primer. Okay, if you want to get more stringent, you can do here instead of 100 to 50. That means it will try to design primer which have less than 1 degree centigrade there. Okay, then you will go to advanced setting where again you can change everything as per your requirement. But if you are beginner, if you don't have much knowledge about all these terms, You should not alter these any components. Okay, you just click anyone and first read about each and everything that what it is corresponds. If you feel like you know that this parameter will be helpful for your primer designing, then only change that. Okay, so now we again go to downwards. Then here you can see there are three types of option present: pick left primer, that is pick forward primer, reverse primer, and hybridization probe. As we were discussing real-time PCR, we get to know that there is a TACMAN probe. Okay, so this is the same thing. If you want internal oligos, then you can pick also pick primer. Even if you have one primer available with you, like you have forward primer, but it's reverse primer, whatever you made in past is not working properly, you can do a single primer also. You can uncheck whichever primer you don't want, and keep check on the primer which you required, so it will provide same thing. Okay, once you are done with everything, you click on pick primers. Okay, it will take just 1 or 2 seconds, and it will give you something like this. Okay, so it have given you first, it always provide 5 set of primers. Okay, first here, then next primer list, then next primer pair, next primer pair, and you go like this. It provide 5 to 10 whatever primers are available. Okay, so first we will go to first primer pair. It is showing that this is the forward primer, this is the reverse primer. It starts as 214th base pair of your original sequence. Its length is 20 base pair. Its melting temperature is 60. Okay, its GC content is 55, and these are the secondary structure predictions. It's okay. So similarly, you can check for reverse primer. As I said, that there should be not more than 1 degree or 2 degree or 3 degree temperature difference between forward and reverse primer. It is showing there that TM of both are equivalent. But you can see that here is a secondary structure in form of hairpin is present. As you can see that this is a 5 prime end of your primer, this is the 3 prime end of your primer, and in between it is getting a bond and making a hairpin. But the TM of hairpin is less than your TM of your primer. So by the time it reaches an annealing step, this should get dissolved. So you can consider it. Similarly, it is telling about self dimer. Whatever the self dimer is present, you can see the temperature here. If it is very low than your annealing temperature or melting temperature, then you can consider this. Okay. So now, for example, we are saying if you go below, then you can visualize the actual position of your forward and reverse primer. This purple sequence highlighted is your forward primer, and this one is your reverse primer. Okay. Now, if let's consider that we have designed primer, and now we want to check its quality. So what we will do, we will go for go to NCBI, you click on Oligo Analyzer tool. Okay, and you can also side by side open Oligo Cal C. Okay, these both softwares are specific for checking your quality of your primers. Okay, for Oligo Analyzer tool, you need to sign in for or register for first time. You can save your password and use it. It's a free software. It won't charge anything. Just you have to um, use for just you have to register for once. Okay. Oligo is not working. Okay, so here algae calculator is there present. So what we'll do, we will copy first one by one. You have to do one by one. First, you copy your forward primer. You will paste here, and it will give you general information about your primer. That what is the melting temperature, which you already have gotten from here. It is already present here. Okay. It is taking a little time. Meanwhile, we can do for pharma designing also for one human sample, because in validation phase we are going to do check on human also. Okay, so let me do one thing. Go to main. You again keep everything same. We just click on pick primers. It will take a little time. Okay, so this is the home page for Oligo Analyzer tool. What you will do, you will paste, sorry, you will take a sequence, whatever primer you got, you can go here and you can paste your forward. First we are doing for primer, then we will go for check hairpin analysis. It will show you all the possible hairpin which are going to form. Okay, you can see that it is showing hairpin, proper hairpin here. This is the structure of hairpin. You can see this is 5' end and this is 3' end. Once you get your hairpin, you will check its melting temperature. Again, you can see its melting temperature is 34. That means it will get dissolved by the time it reach annealing step. And then we will check the self dimer. Self dimer is present there, but its delta G is minus 9.28 kilocal. In case of a self dimer, if delta G range between minus 3 to minus 6 kilocal per mole, it is considered as okay for your primer. Okay, so you can go with this primer synthesis. Now we can, what we can do, we can check specificity using primer blast. Okay, primer, simply Google primer blast. It is primer designing tool for NCBI. First, what you will do, you will provide SSN number or full faster sequence for your sequence, which are you going to check specificity. So we already have our SSN number with us. So what we will do, we copy it, we paste here, and now we will paste our own primers which we have designed. This is the forward sequence we did. So we paste here forward sequence, then we take reverse from here, we paste it here, and we will not change anything, and we just get primers. Okay, it will take a little time. You can uh, get primer for one template, and you can make it for multiple sequences in one go. There is an option in there. Meanwhile, it is get done. Let's directly move to our this In-Silico PCR. Okay, for In-Silico PCR, we have three software. One is UCSC In-Silico PCR. Second one is In-Silico PCR amplification from EHU, and third one is In-Silico PCR tools for this primer distal. Okay, UCSC-PCR contains already genome sequences for all the animals, sun, fishes, and some insects also. Okay, so if you are working with animal tissue or any human tissue or whatever, if your target is there, you can work here. Okay, if your work is on prokaryotes, then you have to use this in silico.ehu.es1. It contains genera. You have to select genera and then you have to paste your sequence. I will show you one by one. And again, in silico PCR tool, this is a very widely used and very easy to use tool. You can run it on a stand-alone mode, that you can download it a exe file and you can run it on your PC with large data set, because online there is a limit of data uploadation. Okay, so you can run it on your PC, you can either Windows PC or Linux PC or Mac PC, but it runs on JavaScript, so sometimes you have to you can run into some issues, plus you have to know how to run that. Okay, so first what we will do, we will go UCSC, we select human genome, again we go our primer file, we take our forward sequence and we paste our forward sequence here. Then we will go to reverse. We again copy and paste our reverse sequence. Okay, now it is asking that how much maximum product size it should consider. Minimum, how much mismatches we can allow, how much good match, minimum good match, and whether we want to flip reverse primer or not. Okay, we are not changing anything, we are directly submitting it. Again, it will check on long data, so it will take time. Meanwhile, we get the result of our primer blast. Okay, so the gene which we are used for primer designing in case of human was vascular endothelial growth factor A, that VEGFA gene. Okay, so here you can see that our primer blast shows that this is the sequence. Hope it is visible. The blue line, primer one, and with two triangles, it is our primer sequence. It is showing that our primer get binds here somewhere in this, between this region, and what gene it is, as you can see here, it is written that it is VEGFA gene. Okay, and further information is already present here. You can see, you can scroll down below, you can see that this is forward primer, this is reverse primer, its statistics is there, what is the length, what is the start, what is the stop sequence, this is all detail, plus where it binds. Okay, so with this, if we are targeting, for example, we are targeting this gene and it is showing that it is binding on the same gene only, so it proves its specificity, that it will not get bind on any other gene or any other off target region. It will bind only the region which we have been selected. Okay, so this is for specificity, or you can visualize. And if you are presenting your work somewhere, if you want to look at more interactive, we want to look at more polished, you can take a screenshot from here and paste in your PowerPoint presentation or your report. It will also look good, plus it will proves that whatever the primers you have designed are actually the reason for the same thing. Okay, so this is the, as we know that this was for primer blast. Okay, now hopefully this is the result from UCSC that we get from, this is the region where our primer get binds. Whatever is capital is our start primer, this is our reverse primer, and this is the target region. What if you are looking for SNP, you can use same sequences and then align them, and then you can mark the region which have SNP. This works very well for that purpose. Next, we will go to In-Silico tool. Okay. So, for example, we will take an example file from here. This is a sequence, our target sequence. This is the primer list is already there. Okay. So we set all the parameters, minimum amplicon size, what will be maximum, how much mismatches are available, and then what we will do, we click on run. Once we click on run, it will again generate same type of things. It will provided the sequence was 4,315 nucleotide base pair. This was the sequence. This is how it binds, and this is the sequence where it get binds. Okay, so you can export this result also. So these are the some simple in silico methods where you can do the in silico PCR before running your actual PCR, so that you can be sure that whenever you are going to run PCR, your primer is actually going to amplify something instead of some artifact or giving you zero results. Okay. Plus, you can use this as a your presentation or your reports. Uh, with this, I like to conclude the today's presentation. Kuldeep, are there any questions? If there are questions, please let me know. So one question is that which PCR is best for detection for pathogen? Um for pathogen, mostly if you are targeting as there are multiple factors involved. Like, sorry, if you are going for this there is a term signature sequence. If you are going to identify a pathogen from a wide set of other pathogens or same genre of pathogens, then sequence first you have to do this what we call it signature sequence identification, that is specific to only that subspecies or species of pathogen. From there you can go with conventional PCR, you can do colony PCR also, but in normal cases nested PCR is always considered as the best, because more than one primer can be run at a time and you will get a number of specific set of genes which you can confirm present in particular pathogen. So nested PCR is better. How to make PCR more affordable for field use? Sorry, till now there is no method other than using tag DNA polymerase. But nowadays, production of tag DNA polymerase due to major players are getting diluted, and many local players are involved in this polymerase production companies are there, so it is getting cheaper. But this is not as much as cheaper than your deep stick method. So there is no certain method that you can say pinpoint that by doing this PCR you can make it cheaper. One is to identify putative function, you can use combination of computational and experimental method, primarily by analyzing sequences. I don't know whether it is a question or someone commented on that. Yes, we can do analyzing sequence, structure, expression patterns, and homology to other genes. They can be done, but whatever you have suggested, it is purely bioinformatical. It is not wet lab. What we are going to do here is combination of wet lab as well as dry lab. So this is, and question is, how do you modify primer design to GC rich or AT rich regions? You can certainly do your whatever your sequence you are taking, as I shown that you have to use inclusion and exclusion criteria in primer 3. You can use that, and then you can include primer 3, or whatever software you are using, you can do the uh, this inclusion and exclusion of your GC rich. One question has been asked that uh, what are the, sorry, I'm not able to read. Can we amplify DNA sequence? There is some glitch in chat, so I'm not. Kuldeep, can you read this? Someone have asked question. What are the latest? I'm not able to read this. Yeah. What are the latest advancement in future direction of PCR? Okay, there are many latest advancement like in RT-PCR. People are trying to reduce the quantity used for PCR. Like previously, it was basic rule that you have to use 10 microliter of reaction. Now it reduced to 1 or 2 microliter. Also, there is a one latest technique that is known as digital PCR. That is the latest advancement in PCR industry, where your template is get encapsulated in droplets, and a very minute amount of your template is required for PCR. Can we amplify DNA sequence if I don't have template strand? If yes, how? Sorry, I don't get what you mean by if I don't have template is strand. Template is called as your DNA, whatever the DNA in real life, that can be used as template. But sorry, I'm not sure. It is from Richa. Richa, please, can you message your question to Kuldeep or your community, whatever, so that I can answer it properly, because it is quite confusing that what you actually mean by that question. Someone is said, okay, someone have already answered the question for someone that yes, if you are beginner, you should not alter the settings in any software, because it will create lots of nuisance in your design PCR, and it will not work well with you. Okay, so if you are not sure, please, plus there are lots of tutorial available in NCBI also, NCBI Blast. If you go there and search for PCR primer designing tutorial, there are lots of tutorial also there. You can question your answer question if you are participating in this workshop and you want designer primer. You can always contact us at Flavinlabs. We will help you without any cost to design a primer. We can guide you if it is not taking much time. Someone is asking, can you brief primary design for CRISPR genotyping or SNP detection? Um, Sorry, that cannot be covered in such less time, because today's workshop is totally for basics of your primer designing and PCR. We can certainly cover this on some other workshop. Which PCR is good to see our gene target gene? If you are going for gene expression, then it is always real-time PCR or qPCR. In crime scene, the amount of DNA, because we don't know about DNA, what we can primary exactly according to. In case of forensics, and especially in crime scenes, there are already primers available which are known as that marker genes. Okay, marker primers, or they are known as marker, simply marker. They are always used. They are specific to human, and whatever is specific to human samples. So the forensics don't design primer every time. Sometimes in certain cases, if there is requirement is there, then only primer are designed, but mostly they are pre-designed primer which are being used in forensic science. Yes, I have skipped about Tachman chemistry and oligo with quenchers. how to design it, because this workshop was only targeted in 1 or 1.5 hour, because our representative or have run this what is a poll that how long you want, and mostly we have seen that after 1 or 1.5 hour people get tired and they starting lefting this workshop. So we are not covering, but if we are interested in specific to RT-PCR, we can then again, and we can do a workshop is specific to real-time PCR, is specific to CRISPR only. Okay. So, sir, you have taught about primer design. Okay, we have discussed this. Which PCR is most useful for COVID detection? As we all have gone through COVID, we saw that for two type of tests was performed. One was your antigen test. Again, second one was your real time, because real time was very specific and very easy to see. Plus, in terms of the result was getting in terms of quantity. So for COVID, your quality real-time PCR was being used. Oligo calculator is free software. Yes, it is a free software, and it is you have to just register. Any student can register. You don't have even provide your all the details. You very bare minimum details you have to provide, and you can register. How to design primer based on restriction site. Again, you have to include and use the inclusion and exclusion for the restriction sites. You have to identify your restriction and jam sites with help of softwares, and then you can provide this restriction site primer. Someone is asking that I want to join your lab. Thank you for your interest. Please send your queries to our official website, and you can just get through that, and we can see how we can do that workshop. Okay, we can do workshop on real time, and if you want to join offline, you are always welcome. You can coordinate to Kuldeep or some person in lab, and you can always join. Okay, so if anyone is still there is query or whatever it is, if you want to talk or you want to discuss something, please free to contact. Feel free to contact our the representatives or myself. I have provided my mail ID and phone number and at last of PPT. So you can always feel, it doesn't matter that you are paid or not for certificate. So if you have joined this workshop, you are welcome to discuss anything anytime with me, or you can come here and do the things. Okay. With this, I would like to conclude this workshop. Hope you have got something to learn and you need to work on that. Okay. With this, I like to conclude, and good luck to everyone. Happy learning, and goodbye.